Many large data compendia on high-throughput genomic and regulatory data have been made available by international research consortia such as ENCODE, TCGA, and Epigenomics Roadmap. Croco provides a network-oriented view on the available data, a simple way to access and browse available information, and a method to combine the data across compendia, experimental techniques, and species. In the following, some use cases of the croco-web application are demonstrated. The web service gives a first view on the networks and enables several network queries, without the need of installing additional software. Croco-web consists of 3 analysis tools: Network Metric Browser, Evidence Lookup, and Gene Set Overlap Browser. In the Network Metric Browser, networks can be selected, combined and compared according to the size, to the network degree of a specific gene, or the overlap with a user-defined regulatory subnetwork. Finally, the results of the network comparison for the selected and combined networks are visualized. In the following, we select several context-specific regulatory networks for mouse ES and T cells. We compare these networks according to the overlap with a regulatory subnetwork, specific for embryonic development. First, we select different ES cells and add them to the selection list. Next, the ES cells are assigned to a group. After that, T cells are selected, and again assigned to a group. The motif overlap metric is selected for network comparison. In the new window, the predefined embryonic stem cell subnetwork is selected. The network consists of 4 genes and 13 regulatory interactions. The results of the subnetwork comparison are visualized as box, line and bar plot. The plot shows the fraction of overlapping interactions of the embryonic stem cell subnetwork in the derived T and ES cell networks. A clear difference in the overlapping interactions between the selected ES and T cell networks and the selected regulatory subnetwork can be observed. The Evidence Lookup tool allows investigating regulatory evidences for user-defined transcription factors and targets. As an example, regulatory evidence for MYOG and MYOD1 in mouse is queried from the Croco network repository. The Prediction and Experimental Binding Site panel shows regulatory evidence for this regulation in TFBS, CHIP and open chromatin data. Furthermore, the Found Literature panel shows literature related to this particular regulation. The Gene Set Overlap Browser allows to browse the Croco network ontology according to the number of interactions between a user-defined set of genes, and to visualize gene set-induced regulatory networks. First, we select the 4 genes, MYOG, MEF2A, MYOD1 and MYF6. Next, we are able to browse the Croco network ontology according to the maximum number of interactions between these 4 genes. By clicking on the plus symbol, the induced regulatory subnetwork for all networks assigned to that particular ontology node is visualized, and can be differentially analyzed.